Welcome to Catholic Ethics and Challenge of COVID-19, a live webcast series presented by the Catholic Health Association and Georgetown University in cooperation with the Pellegrino Center for Clinical Bioethics. Thank you for joining us. My name is Ronnie and I'll be the operator for today's presentation. Before we get started, I'd like to acquaint you with a few features of this web event technology. At any time, you may adjust your audio using any computer volume settings that you may have. There will be a question and answer portion of the webinar. To submit a question, just click on the Q&A icon on the bottom toolbar of the Zoom platform and enter your question. Please note that today's presentation is being recorded and both the recording and slides will be emailed to you. We're joined today by Brian Kane, Senior Director Ethics for the Catholic Health Association. At this time, I'd like to turn the presentation over to Brian for opening remarks. Brian, the floor is yours.
Good day, and thank you all of us for joining us for the final session of this series presented by the Catholic Health Association and Georgetown University titled Catholic Ethics and the Challenge of COVID-19. Before I introduce today's speaker, let us take a moment to reflect on this prayer called Hear Us and Heal Us. Gracious God, we pray for all our brothers and sisters who suffer in mind, body, and spirit. Give them comfort and make them whole. God of love, you sent us Jesus to live among us, to experience our humanity. He went about doing good works, healing sicknesses and infirmities of every kind. He commanded his disciples to care for the sick, to pray for them, and to lay hands on them. God of mercy, you made us in your image and likeness and endowed us with the gifts of love and compassion. Bless all those who strive to live out the healing ministry of Jesus. Give them hands and hearts to embody your mercy. God of compassion, guide caregivers to be in solidarity with the sick and suffering, for surely all will experience these things. May they be as gracious in receiving care as in ministering to the needs of others. Support and strengthen all caregivers as they respond to the needs of persons who are ill, injured, or disabled, that they may provide peace and comfort. And we ask all these things through Christ our Lord. Amen. We are very honored to be joined today by Professor James Giordano from Georgetown University, who will focus on the topic of ethical trials and tribulations of evaluating new therapeutics related to COVID. Dr. Giordano is a professor in the departments of neurology and biochemistry, chief of the Neuroethics Studies Program, co-director of the O'Neill Pellegrino Program in Science and Global Law and Policy, and special projects advisor to the Brain Bank at Georgetown University Medical Center. He is also senior fellow of the Project on Biosecurity, Technology, and Ethics at the U.S. Naval War College in Newport, Rhode Island, and consulting bioethicist to the U.S. Defense Medical Ethics Committee, currently addressing ethical issues in biosecurity and biomedical responses to the COVID-19 crisis. And moderating today's discussion is David Miller, who serves as the Associate Director for Academic Programs and Administrator for the Pellegrino Center for Clinical Bioethics at the Georgetown University Medical Center, where he also directs the bioethics courses for first and second year medical students. Today's event is 30 minutes. If you have questions for the presenter, please go ahead and enter them in the Q&A module. And now I'll turn it over to Jim and David to begin the discussion. Welcome.
Thanks, Brian. My task today will be easy. I will just be monitoring the question and answers, the questions section, and posing questions to Jim at the end. So I won't take up any more time. Jim Giordano, welcome. Thank you for addressing us today.
Thanks, David, and thanks to you, Brian, and thanks to all of you for tuning in today, and for those of you who'll be watching on recorded issue, thanks as well. I appreciate that. And at the end, I also present my contact information. If questions arise as a consequence of either today's lecture or last week's lecture or one of my lectures before that, please feel free to contact me. I will get back in touch with you. I don't guarantee I can do it that particular day, but I certainly will do that. Today, we'll be addressing the idea of ethical trials, clinical trials. But what are some of the technical and ethical tribulations that are inherent to and arise from evaluating new therapeutics, particularly given the intersection of unknowns? The SARS-CoV-2 that is the element that causes, the agent that causes the COVID virus, is unknown to us in many ways. It is new in human hosts. Its patterns and phenotypes of expression are new. And when we're developing new agents that we have some idea may work against other viruses that are similar to SARS-CoV, this is where those intersections of unknowns really play forward. How do we take a relatively unknown drug whose mechanism may be known to us and apply that to a novel agent, a virus, that is not fully known to us? What will that do in individuals who are infected, both in the short, intermediate, and long term? And so these issues become paramount when considering how we engage not only the phased trial paradigm, but what we do after we've essentially put these things into play. For those of you who are not familiar with the way a clinical trial, a phased clinical trial is run, it characteristically entails a preclinical phase in which non-human subjects and/or in vitro methods are involved to test the efficacy of a particular agent, the toxicity, and first-stage pharmacokinetics. In other words, how does that drug or that particular agent move around that system in the period of time and relative distribution to be able to affect what we call the compartments of interest or of aim. We then move into a phase 0, again, in non-human subjects, characteristically in animal models, to test pharmacokinetics in vivo in a living organism under a variety of conditions, particularly with regard to how those conditions would affect the half-life and what we call term effects in those subjects, short, intermediate, and long term. We then move into phase 1, in which the agents are tested in healthy human volunteers to assess the translational safety and relative dose responses in individuals with regard to primary effects and secondary effects. But of course, healthy volunteers and healthy individuals don't necessarily physiologically behave and respond to agents the way those individuals who are suffering from the targeted pathology do. So we then move into phase 2 trials, which then involves patients with the disorder. Here, we're looking at the efficacy under controlled laboratory conditions and the appearance of side effects that may occur as a consequence of the interaction with the agent in the pathologically born host. In other words, an individual who is sick, who may not respond to the drug and/or to the overall treatment response the same way as a healthy person would. We then move into phase 3, which deals with patients under real-world conditions, what's referred to as ecologically reliable and valid conditions. And what becomes important to understand here is what happens at the bench in the laboratory and what happens in controlled environments at the bedside is not necessarily what happens in the real world. So in phase 3, what we're looking to do is test this for its actual effectiveness in real-world clinical practice. And then we move into phase 4, which is where we then survey the drug and its effects longer term after it has been out on the market and it's getting a broader distribution and broader pattern of use in a variety of different shareholders who are the patient populations for which this agent, this drug, or whatever treatment it may be is designed. And the asterisks are important because what we see is a shift in the individuals who are the responsible conductors of these trials, of this research. In other words, who are the bearers of that responsibility and the stewards of knowledge and skill that are necessary for upholding the technical and ethical obligates of what is involved. We move from basic science researchers to clinical researchers, characteristically that are, in fact, clinical researchers or physicians, to moving to private physicians. And then once we're out there in stage 4, this is private physicians. This is real world. The purpose of these phase trials is to provide, develop, afford both a microscope and a telescope. Let me explain what I mean. The microscope is looking at those things literally on the micro to perhaps large micro, maybe meso levels, and the telescope takes it beyond. Not only what's happening with regard to cells and systems, but understanding that those cells and systems are embodied in individuals that are nested and embedded in their environments and their ecologies. And then what are the longer term effects once we get out there in the real world? So if we're to sort of strip this down to basic nuts and bolts, what we're asking when we're engaging this type of research, or almost any type of research, but certainly research that is clinical and translational, is what works and in whom, and under what conditions is it working in those individuals, those subjects for whom it is effective. But it's equally important to extract from this research what doesn't work and in whom and what conditions, and then to compare what's working and what doesn't work to be able to then address and answer the question, why? Why do these things not work? And what does this then mean and inform? What does this mean about the pathogen that we are treating? What does this mean about the treatments that we are developing? What does this mean about the interaction of that pathogen and that treatment in those hosts? And then what does this tell us for the way we're going to utilize these drugs, this agent, in practice? This is the process that engages research in its finer granularity. And of course, what we're looking to do is to utilize a state of the art, what has been described as the sine qua non, there is no parallel, to a form of research approach, which is referred to as the randomized controlled trial. This is neither the time nor the place to get into the real nuts and bolts of what goes into a randomized controlled trial, but I think that the word says a lot. The word randomize, we're randomizing subjects into treatment and control groups. It is controlled. There is some control, whether it's a comparative effectiveness trial where we're looking to then use a new agent, comparing it to what may be the standard of care. And with COVID, that becomes an interesting premise because what is the standard of care? Is it simply palliation of signs and symptoms of those individuals who have actually caught the virus? Is it perhaps engaging some immunological response? Is it a direct antiviral or some combination of both or all? And the question then becomes as to whether or not the randomized controlled trial is actually a gold standard or can be also fool's gold. Here I refer you back to a series of papers that I did with my colleague at Williams College, Dr. Julia Pedroni, where we really looked at the idea of what the randomized controlled trial was intended to do and what the randomized controlled trial may do, may offer, or may in fact inhibit or impede when one is looking to develop precision medicine and or personal medicine. The question here is, how precise can we be in the randomized controlled trial? And or how personalizable can we be or should we be in the randomized control trial? And one of the issues that comes up and is coming up once again repeatedly for COVID is, are we seeing some diffusion in key characteristics and attributes of subjects vis-a-vis patients when we randomize them? In other words, might there be something that speaks back to these questions? What's working in whom? What's not working in whom? What are those conditions, both inherent to those individuals' physiologies as well as their ecologies? And then what does this then tell us about developing treatments that be far more precise with regard to genotype, phenotype, the interaction of genotype, phenotype, and the pathogen, as well as those ecological variables when these things come into real world practice? Well, one of the issues that arises is whether or not there may be identifiable attributes in subjects, again, who may be potential patients, and if those should, in fact, be the target. Ordinarily, in a presentation like this, I would yoke this to some of the work that my colleague, Professor Kevin Fitzgerald, now at Creighton University Medical Center in Nebraska and Phoenix, Arizona, is doing with regard to genotypic specificity and the idea of pharmacogenetics. Can we develop genetic profiles and phenotypic patterns and profiles of individuals to help us identify what might work better, what might not work, and why, and then develop agents and therapeutic methods that are going to be more selective for these identifiable attributes? This is what the researchers Caspi and Bell about a decade and a half ago referred to as attribute treatment interactions. That there may be individual and or group factors that affect positively or negatively various treatment outcomes and results. And my work with Julia Pedroni has suggested that that doesn't necessarily throw out the baby in the bathwater of the randomized controlled trial, but rather, can we just get rid of the dirty bathwater and keep refreshing the clean and certainly keep the baby? In other words, can we do certain things in randomized controlled trials that build in a higher specificity that is then increasing its utility and obviously its beneficence, decreasing its maleficence, and ultimately may afford it greater ethical justice to determine who should get certain treatments, what treatments should be prioritized, in whom, for what reasons, under what conditions. So what this really is leaning to is the intersection between the technical aspects of clinical trials and the ethical aspect, both of the trial mechanics itself and what the trial then infers implicitly or explicitly for its translation into practice. It's addressing the question, who will be helped, who might be harmed, and the larger question is quare, which in Latin is by what means, but for those of you who may be Irish Gaelic speakers, that exact same word means this is huge, this is peculiar. So you've gotten yourself into a quare mess. What is this? What is this, if you will, can of worms or Gordian knot that we need to untangle? And this then speaks back to the idea of what we discussed last week. Surveillance, information, big data, the probity of such data, the importance of these data in helping us to design trials, and then what we do with these data after the fact. But there is an urgency that's going along here. There is the urgency that this may be, quote, a limited time to be able to develop these treatments before the virus then begins to change or integrates itself across what's called a balanced polymorphism in the population, where there are multi-subforms of this virus. And as these multi-subforms express, we see wave after wave of pathogenicity that then perpetuates the pandemic on a variety of levels. On one hand, there have been those that have banged the drum and says, well, why don't we just let that happen? Because what this might then do is impose some level of herd immunity, but not necessarily. Because what we're now learning about the SARS-CoV virus is that this disorder that we've termed COVID-19 may essentially be the stepping stone to other forms of expression, other phenotypes of pathology, COVID-20, COVID-21, COVID-22. And these various viral forms and their expression in different phenotypes of different hosts and populations of hosts may then create opportunities for immunological compromise or reaction that may then pose an entirely new set of clinical issues and problems and burdens. So there is something of a hurry up, bang the drum, let's get treatments out there that are able to effectively mitigate, if not prevent, the onset of signs and symptoms and ramifications of the SARS-CoV virus, whether it's in COVID-19, COVID-20, or COVID-21. Is there some way of expediting the process? And in fact, there is. Once again, this is not the time, nor do we have the time to discuss the full FDA approval process. But there are a number of steps by which, in going through the clinical trial phases, the FDA will then evaluate the relative efficacy of a drug and then approve that drug for constrained markets or broadened markets. And there are certain issues there where the drug may be approved for use prior to a broad market release. The idea here of a pre-market approval, particularly for an investigational new drug, whereby there are no better treatments, quote, out there. And I think with SARS-CoV and COVID, in whatever one of its iterations may be, the question is exactly that. Is there anything else that's out there? If we're looking at comparative effectiveness research, to what do we then use as a viable metric against which to compare something new against that which is out there? And at this point, any new drug represents something that is investigational because we simply don't know how that drug will work against this virus in those individuals across their genotypes and phenotypes who are affected. But if in fact what we're looking to do is bang the drum of hurry up because we do have a limited time to get this offer of benevolence into practice, we have to also at least understand, if not address and consider and take on the contingencies and responsibilities of this expedience. We have to consider the consequences of rushing something to the market or not rushing it to the market. Can we afford to hurry up and wait? Hurry up with regard to getting the thing through phase 0, perhaps into phase 1, but then waiting before we go to phase 2 or phase 3 because we just don't have enough information that allows us to fully inform patients? And what about the veridicality, the truthfulness of our level of concern about what might happen to those who we treat? What considerations are we going to have for the continuity of research and clinical care, and to what extent will that clinical care be provided? In discussions about this topic that I'm having with my international colleagues, most notably those from Germany, one of the questions that comes up is, well, is the social system, our civic institutions prepared, number one, to accept the burden of responsibility to be able to move something rapidly from bench to bedside to broad use? And if not, is there some mechanism for quick change to be able to say that given the contingencies and exigencies of a pandemic and its multifold impact on our biologies, our psychologies, our socioeconomics, might we be able to then develop some system of support that allows a relative, quote, fail-safe? That yes, we're able to get things out there, move them quickly from efficacy to effectiveness, but we're also providing a relative net under the wire that we're asking people to walk so as to be treated for this virus or even vaccinated against this virus, but we don't necessarily know what are the intermediate and long-term effects. So the question then becomes, if in fact we are boldly going, the issues of what those contingencies are, what the exigencies are not esoteric. They really determine in many ways the patterns of response, and they inform our incentives for prioritizing the push of any particular treatment into the market space. And so what we have to do is revisit the balancing act that we've spoken about before. When we put something into balance, what are the decisions that precede our actions? And our group, working together with that of Professor Dr. Nico Kohls at the University of Coburg Applied Sciences in Coburg, Germany, and my colleague Diane Deulis of the National Defense University, have examined something called the Budak Loop. And the Budak Loop is a paradigm for making particular decisions and being informed by their consequences. I mean, this certainly spans from almost the cellular level to the socioeconomic, the way any organism makes decisions. We make particular observations, and those observations in this case are the fostering from research that then orients us to what might be particular applications and decisions to resolve equipoise, to move forward with one treatment versus another. That drives us to the actions, and those actions, what we're looking to do, an intent of those actions is to develop positive consequences and limit negative consequences. And those consequences then feed into our biases about what we're looking to observe and whether our decisions and actions have been correct. And this idea of this Budak loop certainly also takes into account the meanings and implications of the consequences that our actions incur. In other words, we must be responsible for the consequences of our actions. Well, one of the ethical issues that arises is how much information we have. Look, ethics is based upon fact. And good ethical decision-making and action is predicated upon having at least as many facts as conceivably possible, inclusive of the fact that we might not have all the facts. And what's important to guide us here is that a certain aspect of this type of reasoning goes along with ex ubantibus thinking. In other words, because the thing is working, because the treatment is working, and we understand the mechanism of the treatment, we're thinking that's how it's working on whatever the pathogen or whatever is the object of its activity. And there's much ex ubantibus reasoning in medicine, and that's not problematic. But there's also a thin line between what is ex ubantibus aspect thinking and the post hoc fallacy. In other words, it seems to have worked. We've gotten a result, therefore it works because of this. What's essentially known as post hoc ergo propter hoc. In other words, it happened, therefore it happened because of this. But more than that, we have to consider the nature of those things that are happening, not just in terms of the desirable effect, but what might be unanticipated effects that arise as a consequence of the changes that we incur. And this is why the ethics that are focal to this endeavor must encompass and entail the entirety of what's called the ecology of use. I like this word, ecology, and I like human ecology. The cognitive scientist and philosopher Owen Flanagan describes ethics as human ecology, a rational accounting of the environments in which we occupy and live. Our environments, our internal environment and our external environments, and a rational accounting of those things so that we can factor in benefits, burdens, risks, and harms, the relative value of these things to individuals and to the collective, and then ultimately how we're going to distribute these goods and services in both the proximate, intermediate, and long term. So clearly here, the issues come into fore of what is benevolence, in other words, what are our good intentions, and of course, the co-obligate of non-maleficence, not wanting to harm, but understanding that, in fact, harms may occur. Harms may occur because we simply don't know the intersection of these unknowns, which then gets us into the idea of justice. Who is going to get these things? And more than that, who is going to get the longer term benefits or absence thereof? So what we're really doing is we're balancing the past with the present and the future, trying to gain some ethical and technical insight to facilitate both a casuistic look backwards and a predictive look forward and say, are we ready to engage these decisions and actions? Ready, set, go? Are we ready? Are we set? Do we have a choice? The question I ask here is one that I don't necessarily have an answer for. Are our civic institutions prepared to boldly go where we may need to go to develop these treatments, if not preventions and cures, of something as profound as the COVID pandemic? If in fact we're going to take particular risks of expediency because it is hurry up, we also may recognize that there may be particular burdens, if not harms, that exist over the horizon. Where are we going? The vista of probability? Well, we're hoping, yeah, that's the green zone, the probability we're going to use something and this is going to be a viable treatment. But in the zone of possibility, things could arise because of the intersections of unknowns. And in that vista of potentiality, we get some other issues. Are we prepared to cover these as well through ongoing research and, of course, clinical care? And this really is the responsibility for going on the trip, appreciating the tripping hazards, and paying attention to tipping points, which then brings us to the Latin phrase of quidem, or certainty. Quidem in this case essentially meaning that, well, indeed, of course, at least what we require is truthful and clear communication and consideration of the extent of the endeavors of ongoing research and ongoing care, or the absence thereof, if what we're asking to do is to recruit individuals or provide these treatments as a viable public good. I may not have the answers for you because those answers have not evolved yet. This is clearly a work in process and in progress. And my hope is that we make progress and do so in a way that is ethically propitious and prudent. I'll now open the floor for questions if you have any.
Thanks, Jim. I will ask, I do have one question for you from the audience. As you had said, hurry up, bang the drum, and you have this microscope versus a telescope. I'm wondering if we take a more telescopic view, a macro view, there's a lot of competition among different nations to try to come up with a vaccine or a treatment quickly, and there's competition to be the first one to have it. And you mentioned intellectual property, and I'm wondering what are the implications for biosecurity into intellectual property and the possibility of getting treatment out to everyone who will need it when nation states are competing with one another and might want to hold on to proprietary treatments?
Yeah, David, that's a great question. It really speaks to the larger issue of what we're talking about when we're talking about global bioethics. Are we prepared to move away from certain forms of nationalism, certain forms of restrictionism, and to be more open, to recognize the benefit to all, be part of the human community? Or are we going to revert back to that sort of regrettable nationalism and those intellectual property ideas that say, yes, we did this, we got there first, and therefore the benefits and profits to us economically are going to be huge, and then to use those in a way that the philosopher Michel Foucault referred to as biopowerful and or biopolitical ways to be able to then create forced dependencies, non-sustainabilities that are not only macroeconomic in their impact, but certainly microeconomic on the level of the individual. I don't know the answer to that, but I think one of the things we're worried about is if in fact there's not dialogue, discourse, and perhaps productive dialectic before we get to this point, once such a thing, a cure, a treatment, vaccine is developed, you then clearly let the cat out of the proverbial bag, and then you're dealing post facto. And we're worried about such things like as externalized medical tourism, lack of support, product viability and quality control, and what the economic basis for that will be with regard to selection, and then what that means for global justice.
I imagine that's probably true even within nation states when you have competitors like competing pharmaceutical companies trying to come up with a vaccine or something before another company.
Absolutely. And we got a very good comment from the group that I had the opportunity to read, that all of these processes that I think are being fueled by either explicit or implicit senses of urgency have also placed tremendous burden not only on IRBs, but also on data monitoring and safety commissions and committees. And the issue there is an important one, because if we're trying to design trials that move away from some, I think, recognition of things like therapeutic benefit, but to actually confer therapeutic benefit, what we have to then understand is, well, what is the rescue in these trials? So if what we're doing is comparative effectiveness, or if we are using a sham, at what point do we say, all right, this particular treatment is effective enough, but now we can then do a turnover design, a crossover design, to then offer it to those individuals? And if that's the case, well, then is that sufficient to validate the data and create power with regard to comparison? And number two, is that early in the process, and perhaps too early in the process, that may then open the specter for a host of side effects or adverse effects in the future, to which we then must be responsible.
Yeah, it's compressing the time of all of this, and that has so
Correct.
many implications. It's
And this is an interesting thought, because these implications and these issues exist for almost any research. But as you very well pointed out, David, when you compress the time, you increase the slope. When you increase the slope, you increase the area under that slope, the area under the curve of those issues, questions, and problems that have to be dealt with expediently.
I have one more question. We have a couple more minutes. We can squeeze one in. Could you talk about a little bit about complementary and alternative medicine, and whether that's being clinically tried or trialed?
We could have an entire lecture just on that. There is interest here. Obviously, this is pull out all the stops, as many as you can, see what works and see what doesn't. And of course, this has opened the window of opportunity to examine those things that are one click off or two clicks off of what we might consider to be the medical mainstream. Some of them range from those things that are very, very focal to science and technology that we already have in our proverbial wicket, if you will. For example, my group has been working with very, very low dose radiation as a means of stabilizing the immune system to prevent against a cytokine storm, which then ultimately leads to the immunological crash that happens in so many individuals, and acute respiratory distress syndrome. But there have also been some very interesting proposals to then examine a host of phytomedicinals and nutraceuticals, for example, fairly high doses of lysine, which we know can be operative in disrupting the viral coding and making them more responsive to immunological assault. So there's been a number of different proposals that are out there that are not necessarily proposed to consider either or, let's do this or this, but a broader, literally more complementary approach that may then be more inclusive in a both and set of circumstances. And I think under these particular conditions, we have to be open-minded to that, but I think that these conditions also produce particular exigencies and I think concerns and anticipations where individuals may be more vulnerabilized as well. And so certain claims would need to be much more scrupulously assessed and analyzed. And David, as you know, I'm finishing out a paper with a colleague of mine named Latia Dukes about neuro-nonsense and false claims in the age of COVID. And some of them are pretty endemic.
Great. Well, we have run out of time. I'm checking my clock here. So thank you very much, Jim. I also want to take the opportunity to thank the Catholic Health Association for co-sponsoring this program with Georgetown University. We've all enjoyed this, and we may be coming back with another iteration when things calm down after the summer, but hoping that things do calm down. Again, thank you, Jim.
Thank you. And to all, stay healthy, stay well, take care.
Take care.
And on behalf of the Catholic Health Association and Georgetown University, I'd like to thank you for participating in today's event. The recording will be emailed and will also be posted on CHA's website at chausa.org, where you can also find the recordings in this series. Today's program is copyright 2020 by the Catholic Health Association with all rights reserved. This concludes today's program. Thank you for joining us and have a wonderful day.